Okay, you could just talk to me.
Okay.
All right. You look very nice.
Thanks.
So, could you just tell me your name and what you
Yeah. My name is Maria Blasco. I'm a molecular biologist and biochemist, and I study telomeres and telomeres as one of the molecular causes of aging.
And where are you from?
I'm working at the Spanish National Cancer Center, which is the main cancer research institute in Spain.
And what is the story with telomeres? I think it's the one thing everyone kind of knows some little thing about, but...
Yeah. I've been working with telomere biology since more than 20 years ago. So I started working with Carol Greider. She discovered telomeres, and we generated the first telomerase deficient mice, and this was the first time that it was shown that really telomerase was important to maintain the telomeres, and that telomeres were important for aging. So when you induce telomere shortening, this can cause aging. Later on, my group has shown that if you are able to maintain telomeres longer, for longer times, this can delay aging and delay age-associated diseases. More recently, we have been using telomerase in order to show whether it has therapeutic effects in the treatment of human diseases associated with aging, like pulmonary fibrosis, heart infarct, aplastic anemia, etc.
So, did you want to
Yeah,
ask?
go ahead. I'll
Is it
interject
clear?
later.
Because
Yeah,
I'm not...
it's great. It's really great.
Yeah.
And I noticed that in your talk you said something about that telomeres kind of time out the cell. Yeah.
Well, we have seen. I'm not sure I understand your question. What we have observed is that in humans it's known that telomere shortening is a determinant of aging, because if there are mutations in telomerase, these people are going to die prematurely owing to different diseases. So in humans, telomere shortening is known to be very limiting for longevity, but we didn't know whether this was a general phenomenon. And this week, actually, we published a paper in PNAS showing that the rate of telomere shortening determines longevity in many different species, from birds to mammals. So it's interesting to think that, for instance, flamingos and elephants, they share this rate of telomere shortening, and they have a similar longevity. So I think this is telling us that telomeres are important across species to determine longevity, not only in humans.
Is it possible today to actually affect how telomeres work? Could you make telomeres last longer in humans?
Yes, we have shown that using a telomerase activation strategy, which is based on gene therapy. So we have shown, for instance, that if you treat, normal mice with these telomerase, you can delay aging and delay different age-related diseases. And we are using now these to treat different models of disease in mice. So we reproduce in mice different human diseases, like, for instance, pulmonary fibrosis, which in humans is known to be associated to short telomeres. And we are able to cure in mice this disease by activating telomerase.
How do you think that it would be okay to use this therapy to extend human life?
For me, that's not a priority. For me, the priority would be to run clinical trials, for instance, with patients that have mutations in telomerase and that are going to die prematurely owing to extremely short telomeres, to see whether this could be a way to cure these diseases. So for me, the priority is really to use the knowledge we are generating for understanding aging in order to treat diseases associated with aging. I understand that there is a lot of interest in extending longevity, but to me, it's not the priority right now. I think there are many diseases that are killing us that we don't know how to cure, and to me, this is the priority.
This could be the doorstep towards increasing human lifespan by a long time, possibly, yeah?
Well, I think, yeah, of course, maybe at some point humanity can think whether aging can be considered a disease, and whether it's worse to delay aging because it's at the origin of many different diseases. But I think right now probably the most urgent matter is that there are diseases that we don't know how to cure. And I can mention, degenerative diseases of the brain, of different tissues. We don't know how to cure these diseases. And I think from understanding aging at the molecular level, we can come up with therapeutic strategies in order to treat these patients. And I think one of these important mechanisms is telomere shortening. And it makes sense that activation of telomeres could have benefit in the treatment of these diseases. And we have shown that in mouse models of these diseases.
That's fascinating.
It seems to me that, I first became aware of the whole telomerase issue probably 20 years ago.
Yeah.
And it seems to me that at that time it seemed like maybe it was the holy grail of aging, but it really isn't it, right? It's just one of many factors.
It's not the holy grail of aging, it's one of the factors. I mean, there is a review that has been very influential that is called the Hallmarks of Aging. I'm an author in this review. It was published in Cell. And there we discuss different molecular causes of aging. Telomere shortening together with DNA damage or genomic instability, we consider them as primary causes of aging because they can trigger other causes of aging like cellular senescence or exhaustion of stem cells. But of course, telomere shortening is not the only cause of aging. We think it's one of the factors that has been shown, I think, quite clearly that is determining aging.
Is there on the horizon some therapy you could take just a medicine and extend your telomeres, or how would that work?
I think there are several groups, even pharmaceutical companies, that are trying to see the benefits of telomerase activation for the treatment of diseases associated with aging.
And the telomeres, the activation of telomeres would occur through blood infusion or oral medicine or
in
injection?
our case, we're using gene therapy as a strategy. Gene therapy allows for the re-expression of genes in the adult organism, and we are following that scenario, but it could be a small molecule, could be other ways to activate telomeres.
There is one factor in your approach that you spoke about today that interests me because it's something that's been on my mind. Because there's always been the concern that continual expression of telomerase could increase the risk of cancer. So what you suggested you're doing, it's more like a periodic infusion to periodically reset the telomeres. So speak to that approach versus the ongoing, what I would assume would have to be genetic manipulation to constantly produce telomerase.
Yeah, I mean, with our strategy is using adeno-associated viruses. These viruses are non-integrative, so they do not integrate in the genome, and they actually are going to be diluted out as cells proliferate. So that's why we think that by using this strategy, telomerase doesn't increase cancer. We have shown this in many different experiments in mouse models. Even if we induce cancer with oncogenes, we don't see that there is more cancer by putting telomerase. And this is probably because these vectors are diluted as cells proliferate. When you have a cancer or initial steps of cancer, this is characterized by increased proliferation of the cells. They will dilute out telomerase, therefore they will not have telomerase to have an advantage.
I know that you're, based on what you discussed before, you're not a big fan of self-experimentation, but one of the people I had on my podcast a couple of years ago was Liz Parrish of BioViva.
Yeah.
You're probably familiar with what she did with self-experimentation. Did you know anything about how she induced the telomere lengthening, or is that kind of proprietary?
I don't know the story, but I don't know her personally. And while I think there is the clinical trials in order to test whether something is working or not. And I am a complete fan of clinical trials. So if you want to see whether something is toxic or not, whether it's working or not, you need to do a clinical trial. And I think that's the safe way to go.
What's it going to take for telomerase to get from phase 1, phase 2, phase 3 clinical trials? Like how far do you see that?
Yeah, I think of course it's going to take time, but I think probably the first diseases that will be treated, I hope, with telomerase activation strategies will be these Telomere Syndromes. Mm-hmm. These are humans that are mutant for telomerase and they have extremely short telomeres and they are going to develop prematurely diseases like aplastic anemia, pulmonary fibrosis, and there is no treatment right now. So probably this will be, I would say, the first scenario where any potential telomerase activation therapy could be tried. And if it works, it doesn't have any secondary effects, maybe it could go to age-related diseases like heart infarct or other diseases. But I think it's important to do all the steps in research to show first in preclinical models that it works, then move to clinical trials until patients can be treated. It's just for the safety of the patients.
How far away do you think this could be, human clinical trials?
I cannot tell, I don't know.
10 years, 5 years?
Maybe.
Next year?
Next year, I doubt it. No, I think in the following 10 years, something like that. If it ever goes, we never know.
All right. Well, thank you.